Hi, welcome to the Roundtable discussion, which is titled Clinical Trial on the Horizon, Deeper Look into Tina's Wish Progress. I'm your moderator, Kevin Courtney. I'll start by describing my personal connection to Tina's Wish. My wife, Ginny Courtney, was diagnosed with ovarian cancer about 5 years ago. And she was a neonatal intensive care unit nurse who had a deep love and interest in helping special needs children. And we adopted 4 special needs children, all of whom are doing very well. In late 2014, she began experiencing unspecified pain around her back, shoulders, and abdomen. And being a good nurse, good person, looked to her internal medicine provider and went in on at least a few occasions to see what was going on, and took general pain medication. And it was early in 2015 that she was diagnosed with ovarian cancer. And because of the delay in diagnosing it and the fact that there were no tests, obviously, and there were no indications that she was particularly susceptible to it, she was in stage 4 when they found it, and she passed away in late 2017. So that's my connection. She had the chance to meet some people and get involved a little bit with the foundation. From the main session, I'd like to welcome back Dr. Dipanjan Chowdhury from Dana-Farber Cancer Institute and Dr. Kevin Elias from Brigham and Women's Hospital. I'm excited that I get to ask both of you a few questions. And I know everyone who's joined the roundtable is very interested in digging into the questions and the research and the path that your work is on. We'll open up conversation in a few minutes to our attendees with questions submitted on the online chat function. Please type your questions in the Q&A box on the right of the screen if you have any. Before we get started, I did want to mention that we welcome your support this evening in many ways. One way to show your support of the Foundation's mission is to go to the Foundation's website and contribute online. Think of all the women in your life and all the women in the world who could benefit from this amazing research. An early detection test for ovarian cancer has the potential to save more than 150,000 lives per year. The address for the Foundation is on the screen. It's tinaswish.org/donate. I just wanted to point out, and I think you've heard this in the main session, that thanks to the generosity of 4 anonymous donors, we have a matching challenge grant of $5,000, which means that our donors will match dollar for dollar contributions up to $5,000. We hope that you will consider supporting the Foundation's mission and this important work to develop early detection methods for ovarian cancer. And to date, we've raised online a total of $3,486. Let me start while I'm waiting for questions, or more questions, with some questions of my own to the panelists. Dr. Chowdhury, you've already touched on your project and the impressive progress you've made, but I think we could all use a refresher. How would you describe your project to a layperson such as me?
Well, I would describe it as like the goal of this project is when anybody goes to get a physical, and gets their blood test done, which gets some measure of your cardiac health, or whether you have diabetes, the same blood drawn, literally the same vial could be used to portion of it could be used to test for microRNAs to give you a probability score, a score of like, look, it looks like you have no chance of cancer right now, or the probability is very low, or you have some little chance and we should monitor you more carefully, which means you come get another blood drawn 3 months later and we see whether it's changed. That's really where we are hoping this would go. And with ovarian cancer, I mean, we have taken the first step, but the goal is to take it, make it more widespread. But I think we have exciting results with ovarian cancer so far.
Thank you. And Dr. Chowdhury, what were the biggest aha moments or the most exciting developments in your work so far?
Aha moments is, I have to say, this is actually true story, is that when Kevin first came to work with me, which was at the time when he was transitioning from a trainee to have his own lab, and he is a surgeon. So he had 60 patients on whom he had actually done surgery. So and this was just his patients. So a small number of them had malignant disease. Some of them just had benign tumors. And we said we were going to give this 60 patients, the 60 blood samples a shot. And with this first very preliminary experiment, we could see that there were 3 microRNAs could really, with 90% accuracy, tell us who had cancer and who didn't. And this was without all the controls we did later, normal and all that. I believe for both Kevin and me, that was the aha moment was when we first got those results that, wow, we have something really potentially important here, and we need to push forward.
Wonderful, thank you. Dr. Elias, a couple of questions. How accurate is your test currently?
So we think that for the general population, the test has an accuracy of more than 99%, and that it's going to not make a lot of false positives for women that don't have cancer. And it's going to pick up the majority of women who do have cancer, knowing that general population walking around, most people won't have cancer at any given time. And so we think that the test will be extremely accurate. We think even within the high-risk population, the accuracy is quite similar. So even in women who might have a baseline risk that's elevated compared to the average woman, the test will be reliable as far as distinguishing those most likely to have cancer from those who do not.
Very good. Second question, what are the next steps or milestones for the project, and how long do you think it will take to reach those milestones?
I think the tricky part right now is that we feel very confident that when a woman presents to the office, we can distinguish the woman who has cancer versus the one who doesn't. I think what we really want to be able to do is to diagnose the woman who's most likely to develop cancer in the next several years, because we know that if you wait until someone's symptomatic to diagnose cancer, the likelihood that they've already had spread of disease is a lot higher. So we really want to be able to predict those women who have those microscopic tumors or the tumors that are too small to otherwise be diagnosed. So now that we're running the samples from the NIH and we're having the prospective study going through Dana-Farber, we're hoping that within the next 2 to 3 years, we should have that data to be able to say how accurately can we predict cancer for the women who otherwise would have never presented.
Thank you. We have a question from a participant, Sean Lanning, and the question is, how does your approach have the ability, or how much does your approach have the ability to improve over time? For example, up to 99% true positives in decades or time to come.
Yeah, I mean, I think the challenge there is that with ovarian cancer being a somewhat rare disease, so the example that I give a lot is that you can have a test that's got 99% specificity, but if only 1% of the population has the disease, your test will be wrong half the time. So that's why the bar is so high for us, because we really want a test that doesn't need a second line test. There's not going to be a CAT scan or an ultrasound, because we know that previous studies that have defaulted to imaging tests have been unsuccessful because the tumors we want to diagnose are too small to be seen by imaging. So we think that over time we can probably get it to picking up 80 to 90% of the true positives. We know, of course, you're never going to pick up every patient, and part of it is that we're not going to probably screen women who are very young. We'll be looking at the women who are most likely to develop cancer. But we think that 80% of the tumors that I see clinically should be amenable to diagnosis early. And so we, of course, would like to be able to pick up virtually all of those.
Those are good numbers too. Thank you. Back to Dr. Chowdhury. What might women see in your test, or when might women see your test in their gynecologist's office as part of their routine screening within the general population?
I wish I could give you a very specific number. So that's one of the reasons why we started, as we were initially, Kevin and I were focused on the general population, but we started to go towards the high-risk clinic because we believe this test its immediate application will be in these high-risk families. And I say this because, even if you're high risk, it's it's very, very important to know whether you actually are going to get the disease or you're okay right now. So we believe the test will first potentially get to the clinic in the high-risk population. And as we see good results there, and obviously resources would be needed to get it to the larger general population, where it's still a low prevalence disease, which is a good thing. So it will take a little longer. But the high-risk population, we hope it can get to the clinic in a couple of years.
Okay, and in trying to identify the high-risk population, what, other than what women presenting at the clinic, do you have any other plans or methods to try to identify that group?
So right now, the high-risk population, particularly in the Western world, is classified according to some of these genes we know, the BRCA gene. So there are 11 genes that are sequenced, and we are looking for mutations or changes in these genes, and then we classify these families as high-risk families. I mean, the most clinical classification is that whether your family history, whether you have your aunts and your mother, your grandparents, anybody had cancer, and you trace it that way, and then you do the sequencing of the genes. But the other interesting aspect we didn't get time to get to is that we expect that our test could actually catch high-risk families. So we have developed it in a way where it's possible that if you think you're high risk, or if there's a chance you're high risk, we could actually accurately try to capture these high-risk families. So that's another potential application, which is not too far away.
Okay, thank you. Back to Dr. Elias. Are there ways for women to participate in your trial in the Boston area or outside the greater Boston area?
There's two different ways for people to participate. Right now, it's being offered through Dana-Farber. So patients, at least in the current version of the study, would have to be connected to Dana-Farber. Although one of the few silver linings of the COVID-19 pandemic is the fact that we've now moved our genetic testing to being done virtually. In fact, we have a parallel study going on at Dana-Farber now. In fact, I just found out that it opened today to allow people to do remote home genetic counseling and testing to see if they might be at high risk for ovarian cancer. And if so, then they would be eligible to participate in the study. So even if somebody is not local to Dana-Farber, you can still, through our genetic testing clinic, get tested to see, or counseled and assessed to see if you are at high risk, and then if you are, potentially to enter into our study. Yes.
Okay, thank you. I'll just stop for a minute and remind participants that if they have questions, they can type them in the bottom right hand of their screen. And I'll also mention that folks can support the foundation in many ways. You can follow Tina's Wish on various social media platforms, including Facebook, Twitter, Instagram, and LinkedIn. And they are at Tina's Wish ! And again, we ask you to help Tina's Wish reach their goal of an early detection method for ovarian cancer with a much greater audience. I guess let me go back to some questions for either Dr. Elias or Dr. Dipanjan. One is, what are the biggest challenges to fast tracking your clinical trial at this point? That be for you, Dr. Elias?
I think the biggest challenge is it's a bandwidth issue. So if we can open the study to multiple centers, it will run more quickly. Running it just through Dana-Farber, even though we're a large volume center combined with the Brigham, it still will take several years, whereas if we can run it at multiple cancer centers simultaneously, we'll get the data a lot faster. I think that's the biggest issue right now.
Yeah, okay.
That brings us to resources, because there are many centers in the country who have, in principle, they're excited about our data. These are our colleagues and collaborators who are happy to participate. But the key is to provide the resources where they can, draw blood and collect it in an organized way and then ship it to us. So that's all which needs more resources. That's where this is so important, this sort of an event.
Thank you. Now, one question, getting back to testing and identifying at-risk populations. Are there environmental factors in ovarian cancer that you're seeing in the research and the work that you've done so far?
So there are environmental risk factors for ovarian cancer. They're not ones that would automatically make someone a high-risk patient per se. They're the relative risk. So for instance, we know that women who have ever used contraception have a much lower risk of ovarian cancer. So ever having used birth controls lowers the risk of developing ovarian cancer by about 50%. Women who have been pregnant have a lower risk of ovarian cancer, and that risk goes down with each subsequent pregnancy. And women who breastfeed have a lower risk of ovarian cancer, and the longer women breastfeed, the lower their risk. None of those bring the risk down to zero. At the same time, a woman who has never been pregnant or never been on birth control would not automatically be in such a high-risk category that she would automatically qualify for screening without some other familial risk. But we do know that those are relative factors that can influence someone's individual risk.
Thank you. And a question about progression of ovarian cancer. Is there anything like a typical time frame between, beginning at stage 1, going from stage 1 to all the way up to 2, 3, 4, and above?
So Dipanjan, I'll handle that one. The timeline that we're trying to intervene on is actually that timeline between pre-invasive disease and stage 1 disease. We think that time might be something between 5 and 7 years, or at least those early stage 1 tumors. The time between stage 1, 2, 3, and 4, the cancer usually has an acceleration. So the time to go from a stage 1, 2 tumor to a stage 3, 4 tumor is usually very short, maybe on the order of 6 months. So really, we want to try to get these tumors during what we think is going to be that long time interval when they're either pre-invasive or at the very early part of the stage 1, maybe stage 1 to stage 2, before they have a chance to spread more widely.
Okay, thank you. Coming back to environmental factors, I'm just wondering at a very high level, are there broader environmental factors like living in a highly polluted area or, being subject to either air or ground-based pollutants? Have you found any connection there?
There aren't any environmental factors as far as pollution that we found to be particularly associated with ovarian cancer, so no. It seems to be much more closely tied to reproductive risk factors or genetic risk factors. So pollution in and of itself, while it can be closely related to other types of cancer, like lung cancer certainly, does not seem to be a major contributor for ovarian cancer.
Okay, thanks. A follow-up question from Sean Lanning. Out of curiosity, without straying into politics here, do either of you wish the U.S. government did or could do anything different in relation to the ability to fast track a testing technology such as yours, either the underlying technology or the testing process mechanisms themselves?
To be honest, I mean, we would wish every government would do more to fast track this sort of technology. I mean, the biggest issue with diagnosis is there is very little corporate money in it. This is not a drug discovery. There's a lot more resources put into drug discovery for obvious reasons. So I think a lot more support from the government on projects like this would really go a long way. I mean, I can say, Kevin and I, we haven't received a single federal dollar so far for this project in particular, and it's not like we haven't tried. So it is considered very high risk, and obviously, which means high risk of failure. But I don't think we can move the dial by taking a conservative approach for things that haven't been solved for many, many decades. So it is absolutely something we wish that all governments worldwide would take a little more seriously and promote.
Thank you.
I think in general, if funding put a fraction of the dollars they put into developing therapies, into developing preventative strategies or screening strategies, we would make a lot more headway. But fortunately, it's not where dollars usually go.
Okay, thank you. I thought I would mention, just as we're taking a breather, that so far this afternoon, we've raised $3,700 online, and the total raise in this event is $43,900, which is an impressive amount. Let me go with a couple more questions, if you don't mind. We have a little bit more time. Do you foresee other collaborations in relation to this project? I think you mentioned earlier that you're working with clinicians elsewhere in the area and in the country. Do you see greater collaboration or different collaborations?
Oh, it's already happening. For the data we presented you, some of it has been generated with samples from Poland, and our actual close collaborator on this, literally the person who's as critical to this as Kevin and I are, is Wojciech Pendler, who's a Polish biostatistician who's based in Poland, but is affiliated to Dana-Farber. And then we have samples from his hospital in Łódź, Poland. And then we also got samples from India. So it's absolutely international. Kevin and I beg, borrow, and steal wherever we can get samples from, because that's the only way to move this forward. And it's been, really the response overall. People are very interested to participate. There's a lot of red tape. You cannot, take human samples across borders. So we have tried to work around that in different ways. But absolutely, this is definitely a global project as far as we are concerned.
Yeah. Okay. And another question: where and when do you find your greatest inspiration for new ideas as you're working through this effort?
I mean, I think as someone who's in the clinic seeing these patients on a regular basis, that's where the ideas come from. I mean, typically when I am speaking to someone who's been newly diagnosed with ovarian cancer, the second question is about their sister or their daughter or their granddaughter and what her risk of ovarian cancer is. And I can't tell you how often that encounter ends up with somebody saying, why isn't somebody working on a test to catch this earlier? In which my response is, I promise I'm spending most of my time, I'm not here working on that very question. But
I was one of those
really,
people 6 years ago.
That's where it happens, yeah.
I can say from the other aspect of it, the intellectual aspect of it, being in Boston is a real boon that way. There's so much technology being developed, and we being where we are, having very close access to places like the Broad Institute, it's a matter of bringing these things together. So like Kevin is seeing these patients as the human aspect, and then for us in the lab and the technology aspect of it, it feels like it's inspiring to be able to just harness these resources here, which really are not available in almost anywhere in the world. So it doesn't take a lot of inspiration to do this, to be honest.
Okay, thank you. One last question, unless participants have more, is, I'm just curious, how did a radiologist and a clinician connect and embark on this collaboration that's been so fruitful?
I'll let you take that one.
All right, I'll give you the real story. First to just one correction. I'm a part of the radiation oncology department. I'm not actually a radiation oncologist. I'm a molecular biologist, if at all. But it really comes because of the environment here. So Kevin's mentor prior to him transitioning into his own lab was Ronnie Drafkin, who was a really good friend of mine, a pathologist at the Farber. And when Ronnie was leaving the Farber to move to Penn for move his group there, Kevin was still a trainee, and that's when we started talking. And Ronnie wasn't really interested in microRNAs. He's a real ovarian cancer aficionado, but microRNA wasn't his thing. And Kevin was interested in microRNAs, which was my thing. So that's really how we got to it, because Kevin really wanted to use the information we had just generated in the lab for something like this. And it's been a very organic and fruitful collaboration.
Thank you. I think I lied about last question. One other question. Do corporations investing in drug therapies currently see your project as addressing a long-term risk if the public health of the population were to improve, even in this relatively rare cancer?
Well, actually, from a prevalence point of view, as the population is aging, we unfortunately expect ovarian cancer cases to go up. So you'd almost think the converse would be true, in that they'd want to focus on cancers that the population is more at risk for as the demographics change. Mm-hmm. I think the issue is unfortunately a little bit more cynical than that, in that it's easier to charge people a large amount of money for every dose of a drug than it is to do preventative therapy. So I think from a company point of view, they have to be looking at really the scale. You're trying to find a preventative treatment that's not going to cost a lot of money, but be able to be offered to a lot of people, as opposed to what typically happens with drug development. You find a very small group of people who can benefit from your drug and then charge a high price.
Right. Or see a large group and use that as a driver to fund the research and ultimate sale of the drug. Well, thank you. I think we're close to the end of our time. I appreciate everybody participating. I will point out once more that we have an anonymous donor with a matching grant of up to $5,000. That is a match up to $5,000. And as I mentioned, we've raised $3,700 tonight. So many thanks to Drs. Chowdhury and Elias for sharing their expertise with us this afternoon and dedicating their lives to working to saving women. If you were moved by tonight's program, we ask that you consider supporting the mission of Tina's Wish. And as I mentioned before, you can look online at tinaswish.org/donate. So I'll thank you all for joining us and hope you all have a lovely evening.